Okay, so we've just seen how a bolus, a bunch of chewed up food, moves down the esophagus through the peristaltic muscle contractions of the smooth muscle in the esophagus. And once it gets to the end of the esophagus, it's then going to be deposited into the stomach. And the stomach is a J-shaped sac, and it's responsible for some mechanical digestion and some chemical digestion. Instead of having only 2 layers of smooth muscle, here in the stomach, we are going to see 3 layers of smooth muscle. So we're going to have the circular and longitudinal layers, just like we saw in the esophagus, and then we're going to have a 3rd layer called the oblique layer, running in the diagonal direction. And those 3 layers of smooth muscle, again, involuntary muscle contractions, are going to knead and twist and smash up all of the boluses that enter the stomach. So breaking those down even further. That's the mechanical digestive function of the stomach. And then we're also going to see our second example of chemical digestion. So in the oral cavity with the salivary amylase, we saw the breakdown of some sugars. Here in the stomach, we're going to begin to break down proteins. And this process has to begin within the stomach because the control of things entering and exiting the stomach is really, really high. We've got a sphincter, a ring of muscle on the upper end, allowing things in, and another ring of muscle on the lower end, allowing things out. And what that does is give us high control over the really acidic juices, the gastric juices that we're going to find within the stomach. But we need that high acidity to be able to break down proteins. So some of the things that the stomach will secrete into the inside will be hydrochloric acid, which hopefully we know, to be highly acidic, a very low pH, and pepsinogen. And when those 2 things react together, when the stomach has secreted both hydrochloric acid and pepsinogen, it's going to make something called pepsin. And pepsin is what is capable of beginning to break down proteins. So break those individual amino acids down those chains of amino acids down into individual amino acids. That requires that highly acidic environment, and that's why that has to happen here in the stomach. Now, those amino acids are not going to be absorbed yet. We're only going to see that chemical breakdown of the protein molecules. The stomach cannot absorb a lot of things. Absorption is not its primary function, but 2 exceptions to that, things that the stomach can absorb, include alcohol and aspirin. And that's why the effects of those 2 substances on our body are so fast acting, because they are absorbed very quickly within the digestive process. So once all of that mechanical digestion has occurred through those 3 layers of muscle, and the chemical digestion of proteins has been started, and we're mixing up all of the gastric juices inside the stomach and mixing up all those individual boluses that have been swallowed, now what enters the small intestine, leaving the stomach, is called the chyme mixture. But we just saw that the juices inside the stomach are highly acidic. So once those things leave the stomach and enter the small intestine, we want to make sure that we are neutralizing that acid really, really quickly. So that's going to happen as soon as that lower sphincter opens and that the stuff inside the stomach is moved into the small intestine. It's going to be neutralized almost immediately. The small intestine is about 18 to 20 feet long, and we probably know that it's not, it's all twisted up so that we can fit all of that area into our abdominal cavity. And here's where we're going to see the majority of chemical digestion that happens. So we're going to have more amylases produced to break down carbohydrates. We're going to see proteases to get more of our proteins down to the amino acid level. And we're going to see lipase to break down fats. Bile is not an enzyme. It's a mostly water with solutes in there, but it's designed to emulsify fats. We remember from our discussion of the lymphatic system that fat molecules are large. They also have this tendency to stick together. And if we have a bunch of fat molecules stuck together, lipase cannot access that. So the bile acts to keep the individual fat molecules separate from one another long enough for lipase to come in and break them down. And then absorption can occur. The small intestine is divided into 3 regions. The chyme from the stomach enters through the duodenum. That's the purple section at the very top. And then the pancreas and the liver, which we'll see next, supply all of those digestive enzymes. So those enzymes that I mentioned before, amylase, protease, lipase, and bile, are not made by the small intestine, but they're just deposited into the small intestine from the pancreas and the liver. We'll see those right now. So the liver, the pancreas, and the gallbladder, they're not parts of the GI tract itself. They're considered accessory organs that contribute to the function of digestion. The liver produces bile, so we saw that for the emulsification of fats. And the pancreas produces all of those other digestive enzymes needed to break down food to be used by the body. Back up a little. Once their chyme mixture has made it through the small intestine, the majority of absorption has already occurred. But what's left over does enter the large intestine. And the large intestine absorbs the majority of the water remaining in the chyme mixture. It's also going to store and eliminate the feces, the stuff that is left over. The large intestine is rich in bacteria, and these bacteria produce vitamins that can also be absorbed here. The large intestine is made up of the cecum, the colon, the rectum, and the anal canal. The cecum is a small pouch that hangs just below the junction point between the small and large intestine. This is where you'll also find the appendix. The largest portion of the large intestine is the colon, and this is where all of that water absorption is going to happen. What's left after the water has been removed then is feces, and feces is brown because of the pigments found in bile, and it's moved through the large intestine similarly to how we saw the bolus move through the esophagus and the chyme through the small intestine, that movement of peristalsis. But peristalsis in the large intestine is a lot slower, and this allows for more time so that we're making sure to absorb the most amount of water that we can. And then eventually that feces is going to push up into the rectum, which will trigger the defecation reflex, which I think you can imagine what happens after that. Luckily, we have 2 sphincters regulating the defecation reflex. The first one is an involuntary muscle contraction. So once the small intestine is full, it'll kind of like knock on the door of that first sphincter and it will open up. And then the second sphincter is going to be under our control so that we can decide when we're going to be getting rid of that. Okay, so that kind of completes our story of digestion. I've got, again, another list of questions. for you guys to look at. And if you are reviewing your notes and looking through and not sure how to answer some of these, I would love to hear from you so we can discuss it more.